Usually, when we talk about a medical diagnosis, there's this expectation of precision. It feels almost like engineering.
Right, like it's a math problem to solve.
Exactly. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, there it is, that's the problem.
Yeah, it's binary. I mean, it's broken or it's not broken. And there's a real psychological comfort in that, honestly. Because human beings, we really like things to be visible. We like them to be neatly categorized so we know exactly what we're fighting.
But then you step into the world of oncology, and specifically, if you step into the world of pancreatic cancer, suddenly that x-ray machine feels basically useless.
Completely.
We're looking at a diagnostic landscape that is, well, honestly, it's murky. It's not a clear, jagged line. It's this shadowy, moving target.
It is the absolute definition of diagnostic muddy waters.
Oh.
And that specific ambiguity, that ability to hide in the shadows is what makes this particular disease so incredibly lethal.
Which is exactly why we are pulling apart this specific stack of research today. For you listening, we are going deep into a groundbreaking 2026 academic paper from the journal Cancer Cell.
Yeah, from Zhu, Li, Zhang, and the rest of their team. It's an incredible piece of work.
It really is. The research team here completely flipped our foundational understanding of pancreatic ductal adenocarcinoma, or PDA. And our whole mission for this deep dive is to figure out exactly how and why a relatively harmless, quiet little cluster of precancerous cells suddenly figures out how to turn into a lethal invading force.
And to really grasp the magnitude of what this paper uncovered, we kind of need to understand the grim reality of the clinical scene here.
Right. It's not a pretty picture.
No, it's not. PDA is notorious. It has a 5-year survival rate hovering right around 10%. And even in the best-case scenarios where a patient's tumor is caught early enough to be eligible for surgery, that survival rate barely nudges up to maybe 20%. Yeah, it is an intrinsically aggressive, fast-moving disease.
Mm-hmm.
But the really wild part, the precursor to this nightmare, is actually incredibly common.
Okay, wait.
It's called PANIN, which stands for pancreatic intraepithelial neoplasia.
Wait. If the cancer is that rare and aggressive, but the precursor is common, are you saying a lot of us just have these precancerous cells sitting in our bodies right now?
Yeah. Most healthy adults have them by midlife. They're sitting right there in the pancreas.
That is slightly terrifying.
I know, but the way they behave initially is completely non-invasive. They spread throughout the ductal system of the pancreas.
Right, the ducts.
Yeah, so if you think of the pancreatic ducts like the plumbing pipes of the organ, these PANIN cells are basically just lining the inside of those pipes. But they don't break through the pipe wall, what biologists call the basement membrane.
Okay, so they're contained.
Exactly. As long as they stay inside the pipe, they don't cause any clinical harm. So the million-dollar mystery that has plagued oncology for decades has always been, how do these benign pipelining cells suddenly figure out how to breach that structural barrier, break through the basement membrane, and invade the surrounding tissue to become lethal PDA?
Okay, let's unpack how they actually solved this. Because the methodology the researchers used is, honestly, it's pure sci-fi. It's a technology called high-resolution spatial transcriptomics.
Yes, StereoSec is the specific system they use.
StereoSec, right. And the best way I can wrap my head around this is by thinking about baking.
Baking. Okay, I like it.
Stick with me. So traditional genetic sequencing is kind of like taking a fully baked blueberry muffin, throwing it into a blender, and running an analysis to tell you the ingredients. It tells you there is flour, sugar, and blueberries in the mix.
But it's just mush. You don't know what the blueberries were.
Exactly. But StereoSec is totally different. It doesn't blend the muffin. It gives you a high-definition 3D map of the intact muffin, showing you exactly where every single blueberry is located in three-dimensional space.
That spatial context is everything, StereoSec allows researchers to look at an intact slice of a pancreatic tumor and see exactly which genes are turned on or expressed in specific geographic locations down to the single-cell level.
It's basically a GPS for gene expression.
Spot on.
Yeah.
You aren't just seeing a list of active genes, you are seeing where they are active. Are these genes firing in the dead center of the tumor? Are they firing at the invading outer edge? Or, in the healthy tissue right next door?
So to figure out how these cells break out of the pipes, the researchers looked at this spatial map. And that map revealed that there isn't just one type of cell here. There are two totally distinct genetic programs running in these cells.
Yeah, by analyzing those spatial patterns, they identified two main states. The first is what they call MP11.
Okay, MP11.
Right. The low-grade, benign panions, the ones just sitting quietly inside the ducts, are running this MP11 program. MP11 is basically the be a good pancreatic cell set of instructions. It maintains a neat, organized glandular structure.
Just doing its job.
Exactly. The cell is doing exactly what a duct cell is supposed to do, producing normal mucosal proteins and maintaining the structural integrity of the pipe.
But then you have the cells that actually break out and invade the pancreas. And they don't just slowly mutate. They shut down that MP11 program entirely and boot up a totally different set of instructions called MP10.
Yes, the MP10 program.
And here's where it gets really interesting. This MP10 program is the core discovery of the entire paper. Because MP10 is not a random, chaotic cancer mutation. It's not a glitch. When they looked at the genes involved in this breakout program, they realized it perfectly mirrors a completely normal, healthy bodily function.
It mirrors cutaneous re-epithelialization.
Which just sounds so crazy when you first hear it.
It does. But to put that in everyday terms, cutaneous re-epithelialization is exactly what happens when you get a paper cut on your finger.
Okay.
Think about how a cut heals. The skin cells right at the edge of the wound, the keratinocytes, can't just divide to fill the gap. They have to undergo a massive behavioral shift. They stop acting like static skin, they activate a highly specific set of genes, and they physically crawl across the exposed wound bed to drag the tissue closed and seal it up.
Wow.
Yeah. The researchers proved that these malignant invading pancreatic cancer cells are basically digging into our evolutionary hard drive and hijacking this exact skin healing program.
I love visualizing this because if you look at the specific tools the MP10 program gives these cancer cells, it's like watching a heist movie where the crew is gearing up, you know?
Yeah, absolutely. The structural changes are wild.
First, the program builds these things called hemidesmosomes. They use specific proteins, things like LAMC2, LMA3, ITGB4. In a normal, healthy cell, these proteins act like microscopic anchors. They literally bolt the cell to the basement membrane so it doesn't float away.
Right, they lock it down.
But the MP10 program alters how they are used. They stop being static anchors and become dynamic microscopic grappling hooks. The cancer cell uses them to grab the tissue in front of it, pull its body weight forward, and then detach the hooks at the back.
And if you have grappling hooks pulling you forward, well, you also need traction underneath you.
Yeah.
So the MP10 program also ramps up the production of specific surface proteins called integrins.
Right.
These act almost exactly like the treads on a military tank, gripping the extracellular matrix and allowing the cell to roll forward without slipping.
Tank treads and grappling hooks.
Exactly. But physically crawling through dense tissue takes a massive, continuous amount of energy. So crucially, the MP10 program also turns on a suite of metabolic genes, specifically things like SLC2A1 and PKM.
Oh, the fuel supply.
Yes. These genes kick the cell's glucose processing into absolute overdrive, flooding the migrating cell with the rapid combustible energy it needs to fuel this grueling physical journey out of the duct.
Okay, wait, I have to stop you here. Because anyone who has studied even a little bit of oncology is probably shouting at us through their speakers right now.
The EMT question.
Yes. I was always taught that cancer cells spread by shape-shifting. They lose their rigid, boxy shape and melt into these squishy, highly mobile cells, something called EMT, or epithelial-to-mesenchymal transition. But you're saying they're crawling like skin.
It's a really important distinction, and it's a major dogma that this paper actively challenges. EMT is very well known. During EMT, cancer cells repress the genes that keep them stuck together, they lose their top-to-bottom polarity, and they detach to float away as single shape-shifted cells.
Like a brick wall turning into sand.
Perfect analogy. A brick wall dissolving into individual grains of sand that blow away in the wind. But here's the critical detail from the source's widespread EMT actually only occurs in about 16% of late-stage human PDAC tumors.
Only 16%. That's so low.
It is surprisingly rare in this specific cancer. The vast majority aren't doing the whole shape-shifting thing at all.
So what are they doing? Just MP10?
Yes. They are doing MP10. And the MP10 program is mechanistically distinct.
Yep.
In MP10, the cancer cells actually keep their rigid, brick-like epithelial identity. They maintain the strong physical connections with their neighbors. They don't shapeshift into a new cell type. They just adopt the crawling behavior of a healing skin cell.
So they move together.
Exactly. They move collectively. They rip through the basement membrane as a coordinated sheet or cluster, pulling each other along, just like skin healing over scab.
So let's do a quick recap of what we've covered so far for you listening at home. Point number 1, benign pancreatic cells run a normal program called MP11. Point number 2, when they turn lethal, they switch to MP10, which equips them with grappling hooks and tank treads to physically crawl out of the ducts.
Right.
And point number 3, they aren't shape-shifting via EMT like we always thought. They are moving as a solid sheet. So, I mean, have you ever thought of cancer not as an alien invader, but as a confused healing process trying to fix a wound that isn't even there?
That perspective shift is the key to understanding this entire disease. It's a misapplied survival mechanism.
But that brings us to the next much darker question. If this skin-crawling program is the getaway vehicle, who is turning the key in the ignition, like, why does a quiet Pannin cell suddenly decide it needs to heal a wound?
Right, if they all have this program dormant inside them, why aren't all those common Pannin cells breaking out right now? What is stopping them?
Exactly.
Well, it comes down to a localized genetic war inside the cell. It almost always starts with a mutation in a very famous gene called KRAS. KRAS, usually combined with some local inflammation in the pancreas.
Okay, so KRAS is the spark.
Yes, that KRAS mutation is the spark. It initiates the Pannin, it locks the cell into a slightly abnormal proliferating state. It's basically revving the engine. But as we discussed, most Pannins just sit there in the duct. They rarely progress to full-blown cancer.
Right, they just line the pipes.
And that is because the body has emergency brakes built in. We call them tumor suppressor genes, or TSGs. In this context, we're talking about specific ones like CDKN2A and TRP53.
CDKN2A and TRP53. Okay, so these are the brakes.
Yes. The researchers made an incredible observation using advanced mouse models. They saw that rare individual Pannin cells actually try to spontaneously boot up the MP10 breakout program. The KRAS mutation is constantly pushing them, urging them to go.
But the brakes stop them.
Exactly. The moment they try to activate MP10, those tumor suppressor genes sense the abnormal, unauthorized activity and slam on the brakes, triggering a severe cellular lockdown. They physically block the program from running.
Okay, so the cell wants to move, it tries to run the skin healing program, but the breakout only happens if the brakes fail.
And they do fail. Sometimes it happens through random stochastic mutations over a lifetime where the gene just breaks down. Or, as the researchers did in the lab to prove this, they can be selectively removed using CRISPR gene editing.
Man, CRISPR is everywhere now.
It really is. And once those tumor suppressor genes are out of the picture, the emergency brake line is cut. The MP10 cells are unleashed to rapidly expand and invade the pancreas.
But, you know, even with the brakes gone, a car doesn't just drive itself. You still need someone to hit the gas and steer.
You need a driver.
Right. And that brings us to the mastermind of this whole operation. A master transcription factor called FOSL01. And to find this mastermind, the researchers use another mind-blowing piece of tech from the paper called ATAC-seq.
ATAC-seq is brilliant. To understand what it does, you have to imagine your entire DNA as a massive, sprawling library filled with millions of instruction manuals.
Okay, I'm picturing the library.
To fit inside the tiny nucleus of a cell, most of those manuals are tightly packed away in sealed boxes. That's what we call closed chromatin. You can't read them. Traditional sequencing reads every single word in the whole library, packed or unpacked.
Which is overwhelming.
It is. But ATAC-seq only maps the open chromatin. It only shows you the manuals that are currently unpacked, sitting open on the reading tables, actively being read by the cell at that exact second.
Which is a massive distinction. You aren't just seeing what the cell could do, you're seeing what it is actively doing right now.
Yep.
And when the researchers looked at the open manuals during this malignant transition, when the cell breaks out of the duct, they found something glaring. Almost half of the instruction manuals that suddenly opened up were the exact same manuals that open up in epidermal stem cells when you cut your skin.
Yeah. And sitting right in the middle of all those newly opened skin healing manuals was the binding signature for a very specific protein family. By digging deeper, they pinpointed FOSL1.
FOSL1.
Right. Transcription factors are proteins that act like managers. They bind to specific sequences of DNA to turn other genes on or off. FOSL1 is a master regulator. In a healthy body, it's the foreman that oversees skin wound healing. It tells the skin cells when to start crawling.
But here, it's in the wrong place.
Totally the wrong place. Here in the mutant pancreas with no emergency brakes, FOSL1 goes rogue. It binds directly to the DNA of the MP10 genes. It turns on the grappling hooks. It turns on the tank treads. It turns on the metabolic fuel. And it activates all of them at once.
Let me give you an analogy to lock all these moving parts together for everyone. It's like having the blueprints for a highly modified getaway car. That blueprint is the MP10 program.
Okay, I see where you're going.
You've got the KRAS mutation, acting as the shady mechanic in the garage, constantly whispering, we should build it, we should build it.
But the guards are there.
Right. You have security guards, the tumor suppressor genes, the TRP53 and CDKN2A standing around the garage, strictly stopping anyone from touching the tools. Then, one day, the guards go on permanent break. The brakes are gone. The moment the garage is empty, FOSL1 steps in. FOSL1 is the master foreman who rolls out the blueprint, organizes the crew, and instantly builds the getaway car, allowing the cell to smash right through the garage door and escape.
That captures the dynamic perfectly, and the researchers proved FOSL1's role definitively in the lab. When they artificially forced FOSL1 expression in dormant cells, it directly forced the chromatin to open, turned on the MP10 genes, and caused massive immediate invasion into the surrounding tissue. It is the absolute driver of this process.
Okay, time for another quick 5-minute recap. Point 1, KRAS revs the engine, but tumor suppressor genes act as the brakes. Point 2, when the brakes fail, the cell is vulnerable. And point 3, the foreman, FOSL1, steps in and boots up the MP10 breakout program.
That's the cascade.
So my question for you listening is, if FOSL1 is the foreman coordinating this deadly escape, what happens if we look at the environment around the construction site? Because cancer isn't just one cell floating in a void, right? It's surrounded by other tissue.
Right. Are these escaping cells acting alone, or is the environment outside the duct involved?
Exactly.
And this is where the story goes from a cellular breakout to a full-blown systemic hijacking. Because a tumor is a complex ecosystem, and the sources reveal that the MP10 cancer cells are actually actively brainwashing their healthy neighbors to help them escape.
Enter the CAFs. Cancer-associated fibroblasts. The researchers used yet another sequencing technique, single-cell RNA sequencing, or cRNA-seq, look at the gene expression of individual cells one by one. And they used this to study the connective tissue surrounding the tumor.
Right, the stroma.
And they found that the MP10 cancer cells somehow induced the nearby completely normal connective tissue fibroblasts into a specific hyperactive state. They called this state MP4.
And here is the truly chilling discovery of the entire microenvironment analysis. When the researchers compared the genetic signature of these newly created MP4 CAFs to known cellular databases, they found they were nearly identical to the specific fibroblasts found in, well, healing mouse skin wounds.
I mean, the cancer is so committed to the bit. It doesn't just think it's a skin wound, it forces the healthy tissue around it to also act like a skin wound.
It's wild. It creates a localized wound healing environment to support its own escape.
Yeah.
But it's actually worse than just mimicry. It creates a vicious, self-sustaining positive feedback loop.
Oh, no.
Yeah. These wound-like fibroblasts don't just sit there looking pretty. They start secreting powerful signaling factors. Specifically, things like CCN2, VTAN, and a major one called CTHRC1.
Okay, so they're pumping out these signals.
Exactly. They pump out these proteins that float over to the invading cancer cells and bind to a receptor on their surface called EGFR.
The epidermal growth factor receptor.
Right. And when EGFR on the cancer cell is activated by the signals from the fibroblasts, it acts like a giant antenna. It receives the signal and sends a message deep into the cancer cell that specifically boosts the production of FOSL1.
Wait, are foremen?
Exactly. The foreman gets a massive boost.
Oh, wow. So FOSL1 turns on the MP10 breakout program. The MP10 cells then brainwash the local fibroblasts to turn into wound-like CAFs. Those CAFs secrete signals that hit the EGFR antenna on the cancer cell. That antenna boosts FOSL1, which boosts MP10 even more, which makes even more CAFs.
It's an endless loop of malignant acceleration.
To stick with our analogy, the cancer cell doesn't just build a getaway car. It brings its own cheerleaders to the breakout site. The CAFs are basically standing on the sidelines screaming, keep healing, keep moving, and literally handing the cancer cells the chemical energy to do it.
That's exactly what's happening. And this isn't just theoretical computer modeling either. The researchers provided high-level clinical proof in living systems to show how vulnerable this loop really is.
Okay, let's hear the evidence.
They took mouse models, heavily burdened with these aggressive invading tumors, and treated them with specific drugs to try and break the cycle. Specifically, they tested a drug called erlotinib.
Erlotinib.
Right, which is an EGFR inhibitor. It basically acts as heavy-duty earplugs for the cancer cell. They also use rapamycin in some tests.
So if the receptor is blocked, the cancer cell goes deaf to the cheerleaders. Does the tumor just stop in its tracks?
The results were staggering. In just 1 week of treatment, physically blocking that EGFR signal completely halted the invasion of the tumor into the surrounding tissue.
Just 1 week?
Yeah. Without the constant encouragement from the fibroblasts, the cancer cells just couldn't sustain the massive energy required to run the MP10 program. The cells were trapped. The feedback loop was completely broken.
That is incredible. So time for our final quick recap for the listener. Point 1 cancer cells brainwash fibroblasts into MP4 CAFs. Point 2 CAFs sends signals to the EGFR antenna on the cancer cells. And point 3 that boosts FOSL1, creating a deadly loop. So knowing this loop exists, could the future of cancer treatment be silencing the tumor's cheerleaders instead of just attacking the tumor itself?
That is the exact therapeutic paradigm shift this paper is advocating for.
Yeah.
For decades, the strategy has mostly been to attack the tumor directly, to try and poison the cancer cells and hope they die before the patient does.
Right, the brute force approach.
Exactly. But if we pull all the way back and look at this entire intricate biological story, it is a masterpiece of hijacked biology.
Yeah.
A normal pancreatic cell gets a KRS mutation. It sits quietly as a benign pinyon because its internal security guards, the tumor suppressor genes, keep it in check.
But then the guards leave.
Right. The moment those guards fail, the master foreman FOSL1 boots up a literal ancient skin wound healing program. The cell generates grappling hooks to physically pull itself out of the duct, all while brainwashing local healthy tissue into a wound-like state to constantly feed it growth signals through that EGFR feedback loop.
And this is exactly why you, listening to this right now, should care about this deep dive. This isn't just academic trivia about genes and pathways. This research redefines the very taxonomy of pancreatic cancer.
It really does.
It proves that this deadly disease isn't just a chaotic pileup of random mutations. It isn't a shape-shifting alien. It is a highly organized, hijacked tissue repair program. And because it's organized, scientists now have a map. They have specific targetable vulnerabilities like EGFR or FOSL1 to stop pancreatic cancer from ever spreading in the first place.
It changes the battlefield entirely. We are no longer just throwing toxins at a mystery. We are dismantling a very specific mechanical escape route. But I think this paper also leaves us with a profound, almost philosophical, biological question to chew on.
I love these. Lay it on us.
Well, if pancreatic cancer, which is widely considered one of the most lethal, chaotic, and destructive diseases on Earth, is fundamentally just a highly organized, localized wound that never heals.
Right.
What other deadly diseases are actually just ancient, essential survival and repair mechanisms that are simply being triggered in the wrong place at the wrong time?
Wow. Are our bodies just misinterpreting the signals of their own survival? I mean, are our worst diseases just our healing mechanisms acting completely out of context?
It's entirely possible.
That is the kind of question that changes how you look at biology entirely. And I hope it keeps you thinking too. Thank you for joining us on this deep dive into the true nature of pancreatic cancer. Keep asking those questions, keep looking for the underlying mechanisms, and remember to always view the human body's flaws through the lens of its incredible, if sometimes deeply misguided, evolutionary programming.
The muddy waters of oncology are finally starting to clear. But, it turns out we're just looking at a distorted reflection of our own healing.
Until next time, stay curious.